Hello, this is Dr. Panigrai. Welcome to my biology classes for NEET exam. Today, I will be talking about different types of immunoglobulins and their function and a little bit of structure of immunoglobulins. So let's start. What is immunoglobulins? If you observe the immunoglobulin, then such term itself says, what is this? Immuno, it means or it indicates immunity, and globulin, which indicates protein. That's why you can say the proteins which involved in immune system, these may be known as immunoglobulins, right? Yes. The immunoglobulins are the protein which actually secreted by plasma cell in response of antigenic stimulus in the rate of 2000 molecules per second. So these immunoglobulins actually secreted from the plasma cell in presence of or in response of antigenic stimulation. So these immunoglobulins actually composed of glycoprotein. So the immunoglobulins are glycoprotein. It means the immunoglobulins composed of about 96% of protein and approximately about 4% of carbohydrate. So what is the difference between immunoglobulin and the antibody? So immunoglobulin, which provides the structural and chemical concept, but antibody, which provides biological and functional concept. That's why you can say that all antibody are immunoglobulins, but all immunoglobulins may not be antibodies. And these immunoglobulins, which constitute 20 to 25% of total serum protein. And this immunoglobulin, you can write like this, capital I and small g. And these immunoglobulins actually composed of 4 polypeptide chains. Out of these 4 polypeptide chains, 2 polypeptide chains are larger, and these larger polypeptide chains also known as heavy chain, or in short H chain. And there are another two short polypeptide chain, these are known as light chain or L chain. These polypeptide chains are held together by formation of disulfide bonds. So these are disulfide bonds, and the number of such disulfide bond may vary. And this region, you observe that this is the bending region. This region is known as the hinge region. And if this is the polypeptide chain, and it is obvious that it has two terminus. One is the N terminus or the amino terminal, and another is the C terminus or carboxylic terminus. So these are the two terminal, amino terminal and the carboxylic terminal. This region, which forms a Y-shaped structure, this is the structure of immunoglobulin, general structure of immunoglobulin. This is the single unit, and which you can say this is the monomeric or single unit of immunoglobulin. The top of such Y-shaped structure, this region actually bind with the antigen. So this is the antigen binding site. This is the antigen binding site. And you can divide the immunoglobulin into two regions. These are the two regions. The upper half region, it may be known as the fraction of antigen binding site, and the region towards the carboxylic terminus, this half portion is known as the crystallizable fragment. This is known as the crystallizable fragment. So this is the structure of the immunoglobulins. On the basis of physiochemical and antigenic differences, there are 5 types of immunoglobulins. The immunoglobulin A, immunoglobulin D, immunoglobulin E, immunoglobulin G, and last one is immunoglobulin M. Actually, these are immunoglobulin alpha, immunoglobulin delta, immunoglobulin epsilon, immunoglobulin gamma, and immunoglobulin mu. Out of these 5 immunoglobulins, the immunoglobulin M, this is the larger in size. This is the larger in size, and this is pentamer. This is pentameric. It means that 5 number of units of immunoglobulin forms immunoglobulin M. And out of these, the immunoglobulin A, it is dimeric. Dimeric means 2 unit of immunoglobulin forms the immunoglobulin A, and these are monomeric, single unit. These are the single unit of immunoglobulins. The immunoglobulin A, which is also known as the secretory immunoglobulin. These are known as the secretory immunoglobulin because such immunoglobulin present in the colostrum, the first milk of mother, which present in the body secretions like tear, like saliva, like mucus, and so on. That's why the immunoglobulin A is known as the secretory immunoglobulins. The immunoglobulin D, this immunoglobulin actually responsible for differentiation of B cells or beta cells. That's why the immunoglobulin B also responsible for production of antibody. The immunoglobulin E, this immunoglobulin actually responsible for allergic reaction. Allergic reaction. It also responsible for type 1 hypersensitivity. Hypersensitivity, and which also known as anaphylaxis. Anaphylaxis. The immunoglobulin G, such immunoglobulin actually can pass through the placenta. It can pass through placenta and provide natural passive immunity. Such immunoglobulin G also present in the milk of the mothers. The immunoglobulin M, this is the largest immunoglobulin, and this immunoglobulin actually secreted earlier to be synthesized by the fetus. So this is the first immunoglobulin which is secreted earlier by the fetus. And such immunoglobulin also protect our bloodstream. It protects bloodstream. Out of these immunoglobulins, the immunoglobulin G, the percentage of immunoglobulin G is highest in our body, And the second abundant immunoglobulin is the immunoglobulin A, which constitute 10%, and immunoglobulin G, which is the percentage is the 75%. Here, one more thing is that the immunoglobulin G also responsible for opsonization, or you can say the immunoglobulin G also help in the process of phagocytosis. That is very interesting thing. The immunoglobulin G, which indicates the chronic infection, and immunoglobulin M, which also indicates the acute infection. So these are the different immunoglobulins, and these are the different functions of the immunoglobulins. Examination point of view, the functions of immunoglobulin is very important. I think you will enjoy better. If you like, then click on my next session. Thank you for